dementia researcher with a blog and a rating.
The hunt for tiny messengers. Science time! Today we're going to talk about extracellular vesicles. You know, those things that dude down the corridor works on that you don't really believe are a thing. We'll go the semi-patronising route I took with the stroke chat, and I'll tell you a little bit about the history of the field, a little bit about what extracellular vesicles are, and a little bit about why they're interesting. We're going to start in the 40s, where you could get away with writing a series of 30 papers on basically the same thing, each with only 2 beautifully hand-drawn graphs and 1 meticulous table in. There were these 2 chaps, Chargaff and West, who were a biochemist and a medic respectively. They were trying to figure out how all the disparate components of blood came together to make a clot, by spinning down and separating all the components to see what they did. In 1936, they began a series of papers called Studies on the Chemistry of Blood Coagulation, and paper 19 of the series is what we think might be the start of the field of extracellular vesicle research. They found that the addition of the high-speed sediment to the supernatant plasma brought about a very considerable shortening of the clotting time. Basically, in the truly broadest sense, don't hate me, extracellular vesicle people, if you spin down blood with anticoagulant in, you get plasma. If you spin down that plasma, you get extracellular vesicles. And a load of other stuff, but we'll get into that on a later occasion. In the next paper in the series, they took plasma from a normal adult, a haemophiliac, and a female with what is described as a clotting disorder, with no apparent interest in which one. You can tell this is the 30s because the details are sparse and the N is 3. They timed how long it took for clots to form in all of these patients and found, no surprise, the haemophiliac and the female with the clotting disorder didn't clot all that quickly. Then they took the sediment they found earlier, the one I said is probably the extracellular vesicle fraction, and applied it to these patients' bloods, and found that the time to clot was much quicker. Whilst they said at the time that this fraction probably includes, in addition to the thromboplastic agent, a variety of minute breakdown products of blood corpuscles, they did not call them extracellular vesicles or explore them any further. Now, between the 40s and the 80s, there is a ton of literature that never really went anywhere. People seem to have found extracellular vesicles in all sorts of places and not pursued them as anything exciting or novel. If you're interested and patient, there's a history of extracellular vesicle review due out sometime soon, care of yours truly and a whole bunch of much more eminent extracellular vesicle people. But where the field really started to come into its own was in the 1980s. Rose Johnstone and Philip Stahl were looking at receptor trafficking. They both published papers in 1983 on the transferrin receptor. In these papers, they had found the receptor on vesicles within multivesicular bodies and had found these vesicles were released by exocytosis. Whilst the term exosome cannot be attributed to either Johnstone or Stahl, but rather to Eberhard Trams of Ursula Heiner's lab, Rose Johnstone was the first to officially describe exosomes. She hailed them as lipoprotein structures with a phospholipid composition characteristic of red cell membranes. This leads us neatly to modern classification systems and the sticky mess therein. Most people will have heard of exosomes, but it's now considered a rather old-school term, and the more widely accepted one is extracellular vesicles, or EVs, as I will now call them because it's less of a mouthful. EVs can be broadly classified into 2 camps according to their biogenesis. Large EVs, usually more than 150 nanometers, are shed from the cell surface. These used to be called microvesicles or microparticles. Small EVs, usually less than 150 nanometers, are formed by invagination of the cell membrane. They're stored in multivesicular bodies and released by exocytosis. These used to be called exosomes. The problem here is that there is a lot of overlap in sizes and some shared markers between the different classes. ICEV, the International Society for Extracellular Vesicles, has published some great position papers on the issue, so feel free to go and peruse should you wish to know more. That covers what they are, but what do they do? This is where we run into the EVs aren't a thing issue. Despite her initial discovery of them and her continued research into EVs for a number of decades, this problem might have been Rose Johnstone's fault. In a paper in the early 90s, she says, The observations with the transferrin binding are consistent with the conclusion that exosome formation may be a major route by which maturing cells selectively lose obsolete plasma membrane functions. This led many researchers who came after her to believe that EVs were just a way for the cells to shed junk proteins. The EVs were nothing more than cellular debris with no further function. It was the mid-90s when papers began to show that EVs were actually functional. One paper which was particularly important was by Graça Raposo and her colleagues, who demonstrated that EVs from immune cells are capable of presenting antigen. This paper has been described as a watershed moment, catapulting the potential of EVs to be harnessed as therapy. Just over 2 decades later, this is beginning to become a reality. EVs are used in all facets of disease. There are FDA-approved diagnostic tools using EVs, and companies manipulating their chemistry to use them as therapy. But my interest has always been function. Where do they come from and what do they do? And how do they do what they're doing? Let's think about them in the context of dementia, otherwise I feel like I'll lose you to my history lesson. Although, let's be honest, it's still going to be a bit of a history lesson. A search for exosome and dementia brings up papers starting around 2006, demonstrating that EVs are capable of acting as a vector for amyloid beta, that they act as release mechanisms to shed amyloid from the cell, and that this EV-associated amyloid ends up within plaques. But a search for microparticle or microvesicle and dementia brings up much earlier papers from the late 80s showing very similar phenomena. There is now considerable evidence that in a number of neurodegenerative diseases, EVs can propagate misfolded protein in a sort of broken version of the game telephone, but with cells. We know that if we inhibit the release of EVs, we can slow the propagation of disease. We also know that other cells within the brain can take up these EVs, cells like microglia and astrocytes. These cells can become activated, starting to produce reactive oxygen species and reactive nitrogen species, and causing neuronal death by what one of my colleagues wonderfully describes as chemical-friendly fire. EVs are capable of crossing the blood-brain barrier, and the hunt is now on for brain-derived EVs in the blood. Because EVs are so prevalent in the blood, looking for them there in pathology is often described as a liquid biopsy. This is increasingly becoming of interest to those in the field of CNS disease, where catching things like dementia early, before it becomes debilitating, is vital. Biopsies from the brains of otherwise healthy people tend to be frowned upon, but a simple blood test in asymptomatic populations might not be unreasonable and might allow us to jump in early and start something prophylactically. So EVs have the potential to propagate disease, to diagnose disease, and maybe even to be used as therapy in disease. And in challenging diseases like dementia, all of those things could prove important. I think we've established that they're definitely a thing. So if you have time, go down the corridor and chat to that dude about them. You might learn something fascinating.
Thank you for listening. Join the dementia research bloggers and share your own views.